Happy Wine Wednesday, everyone. Cheers to you all. I hope you've had a fabulous week and that your tanks are doing swimmingly. Is that a word? I don't know. But I do have Ray and Holly with me as always, and Sophie, our newest member. And tonight, Sophie is going to take the reins because we have a special guest who she will introduce and I will hush for once. But I do have my Chardonnay. Okay, go on, Sophie. I'm hushing up.
All right, so I think if you followed any of our 3 accounts, Cultured by Science, Canadian Seaquestrians, or the Seahorse Whisperer, you already know who the special guest is. It's Dimo from Cultured by Science. Cultured by Science is making a huge impact in our seahorse world by doing research and by breeding captive bred seahorses. So, Dimo, can you start off by telling us a little bit about your business?
Sure. First of all, thank you for having me. It's a pleasure to meet other like-minded people and to talk about our crazy hobby. So Culture by Science is essentially a extension of my undergrad and graduate studies here at the University of Guelph. We're essentially attempting to run a entirely aquaculture-based business that supplies corals, fish, and inverts directly to the aquarium hobby. Yeah, that's more or less in its essence.
Okay, and so you did all this schooling about it, but why did you choose to go into this?
It's always been a passion of mine. My dad before me had a freshwater aquarium, pretty much as far as I can remember. He also had a fish room. He was a building manager in his very, very early days. And pretty much every broom closet was filled with a fish tank or two. And those were some of my fondest memories. So it took me a while to go back to school after high school, but eventually I decided that I should do something that I really enjoy. So I went to Guelph to study marine and freshwater biology. Because I was already a little bit more mature, I guess, I already knew that school alone was not going to be enough, not only professionally, but to satisfy my thirst to progress in the hobby. So I went to the Hagen Aqua Lab by the university on campus, and I found basically the only tropical coral lab in Ontario for sure, most likely in Canada as well, at least to the scale that we were at. And then from there, I stuck with them. And to some extent, we still have ties with the lab about almost 10 years later now. So okay. I did my undergrad and graduate studies in the same lab.
So why would you say, because I know you've done a lot of studying and you breed them, but why would you say that this research is important for the ocean or for the seahorse community?
The research that I did was basically, I looked more in depth at the aquaculture specifically of seahorses, but also at methods to increase the productivity, overall productivity of a hypothetical system where we would be able to integrate various components such as macroalgae, different snails, urchins, things like that, to create more diverse systems and to also study the diseases that affect seahorses.
Okay, and then,
yeah.
Sorry, has that made like a big difference in kind of what you do compared to like what other people do?
Yes and no. I think that science in general teaches at least the overall scope of, I think, what I would like to bring to the industry and to the hobby with my studies is my or the scientific method and the way of thinking about problems and setbacks. So being very analytical and critical when you do have an issue, and systematic as well in your method for finding a solution. So that's where I think the science applies to the hobby. Um, my research definitely did have some impact. We actually developed, and it still has not been published, the first seahorse cell line. So it's a continuous or immortalized cell line. It's a group of cells that essentially will divide infinitely. Yeah, so that's I'm hopefully going to have some time to sit down and write and finish writing the paper so we can publish it. That would be very, very interesting, because then basically what happens is those cells can be dispersed across across the world, and researchers can use them to study various diseases, bacteria, viruses, et cetera, without actually having to use the animal itself, which means that we're automatically reducing the need for harvesting from the wild or from other captive settings, right? And then the aquaculture aspect, of course, right? It goes twofold, really. The first one is that we're directly attempting to curb any appeal to harvest from the wild. But secondly, the kind of general objective when we're talking about tropical aquaculture is how to find ways to um, bring a relatively simple system to coastal communities. And because we're in some ways taking away their jobs by aquaculturing here in developed nations, to give them back something to do, right? So instead of having to go out and catch fish in the ocean, they can set up these very simple systems and still provide a product that can be then shipped across seas, right?
And be healthier. I'm doing my first interruption. Okay, you guys can number them. Sorry, but we got to put him in touch with Chris Carey, with Graham Short. Graham Short is an ichthyologist who would love to not need bodies. I mean, like, that's his job, but I mean, it'd be cool to get you guys in contact with each other. And then Chris Carey is huge into aquaculture, and I love that you explained why aquaculture is so important. And I think, I feel like I'm echoing. Shut up again. Go ahead, Sophie. Sorry.
So I was just gonna say, it's honestly really interesting to see kind of someone else's perspective in this group of like seahorse keepers, because I know I've only been doing it for a very little bit of time, but also for Ray, Holly, and Kelly, it's more of an experimental sort of thing, less of like a science. But I would say that's really interesting to see. So I know you do seahorses, and you mentioned a little bit of coral. I remember also when I came over to get my seahorses, you had some fish and some shrimp. What else do you do other than seahorses?
So as part of my thesis, like I mentioned already, I tried my best to see what other species are relatively easy to breed, and co-culture, or basically culturing two different species in the same aquarium in the same environment, can be quite beneficial, and have mutual benefit, actually. Specifically, that's kind of what co-culture is. So yeah, the shrimp was one of those animals. So they're particularly, I work with the peppermint shrimp, or the shrimp from the Caribbean or Florida. Its scientific name is Lismata verdemani, and it is quite sought after in the hobby because, of course, it eats. I'm sure you guys know it eats Aptasia, which is the number one pest probably in the hobby itself. So anyway, that's one of the big animals that I'm currently focusing on, and I think that there's also a big difference, at least in my experience, between breeding and trying to bring a species to a commercial scale of breeding, and there's challenges that stemmed from that. Yeah.
Real quick, next interruption. Was the common name of the thing you were talking about the nudibranch?
Yes. Yeah. And
Okay, all right, because some of us are common people. I'm a common person, but I in the reel we put that in because the nudibranchs are really great for eating Aptasia, huge for keepers, huge. They are slow, but I've got tons of questions. I'm gonna wait till it's question time and hush up again. Go ahead, Sophie. Sorry.
All right, wait, I think we should open up the pictures because I know that Dimo has provided some pictures. If you want to do that, Kelly, please. Oh, where's Kelly?
I'm getting them.
Okay.
Sorry, guys. I'm getting them. Okay, so it's taking a second. It's loading very.
Yeah, so I noticed in your pictures you have a very big setup. You mentioned that it was the setup you had while you were writing your thesis. So we'll talk a little bit about that because there were a lot of components and I didn't understand everything that was going on in those pictures.
So the systems were actually, even if they might look quite substantial, they're actually quite small. So the idea was actually to keep all of the setups confined to about 200 square feet, and fill as much of that space with aquariums, of course. But there is nothing really, I believe, that was complicated or that the average person could not do. That was part of the goal there.
Okay, all right, we're ready to share. Here we go. So is this what you were talking about? Let's see. There we go. And then is this what you're talking about? Simple setup?
So that's the first of two grow out setups. So as you will notice immediately, would you like me to just talk about them as we go?
Yeah.
Yes,
Wonderful.
feel free.
So this is one of the like I said, grow out setups. It is a recirculating system. It basically has three tiers. The top one being a 90-gallon surrounded by two 20-gallon extra tall aquariums. In the middle, we have just a 90-gallon 4 feet by 2 feet, along with the 20 on the right. And on the bottom, we have two 29s, as well as just a very simple 20-gallon sump. So everything flows into that sump. The sump is filled with a relatively small, I think it was about 10 kilograms of live rock that was pre-cycled before starting experiments. Just a simple skimmer, some filter socks, and a UV sterilizer. That's one of the big things that pretty much all of my setups will include is always a UV sterilizer. From there, just back briefly, because from this photo you can't really tell, the two tanks on the top that surround the 90 are actually chrysals. So that is something, and I will not take credit for this. It's Zach that does a lot of, or pretty much all of the building, the constructing, and the plumbing, electrical, things like that. He's very, very good with and very handy like that. But anyway, so they're chrysals. So we've made them ourselves. There is a chrysal essentially is a tank within a tank, for those that don't know. And you can't really tell, but there's a sheet of acrylic in both of those aquariums that has been silicone to form a half circle. So the idea is to basically break the surface tension of your regular aquarium. And the reason for that is they're usually used for jellyfish rearing, because the jellyfish, if you ever notice them, they'll always, especially when they're very small, they'll sit at the very surface. And they'll get way too close to each other at a high density, and then they'll start to just make a mess. And the same idea here is with actually the seahorses as well. So when they're first born, instinctively the seahorse will rise to the surface. Why? Because most plankton in wild will be found in the first meter of water. And basically, for the same idea, that's why we use the chrysals. We use the chrysals so we actually submerge them into the water, because otherwise what we found, at least in my experience, is that they clump together. And because we were primarily a disease laboratory, we were always concerned that when one animal gets sick, close quarters means everyone will get sick. So that was
Oh, yes.
that
And
was
Kelly,
the big.
I think there's a picture of one of those tanks. Like I sent you a picture of one of those tanks. Yeah.
There
All
is,
right.
yes.
Well, I'll back up.
There it is.
Is this it? Okay.
Yeah, so this is after many iterations of these chrysals. At first, they were very, very, very simple. Now they're a little more complicated. The biggest problem with the chrysals, well, there's two of them. The first one is that they eventually leak because in the corners where you see there's actually no water there, so there's a lot of pressure that is constantly exerted on the silicone. But at the same time, the more important biological issue with the chrysals is that the mesh that you'll see at the very left by the pipe, by the standpipe, has to be sufficiently large enough as to not create much of a pressure where the animals will then be sucked into. Does that make sense? I don't know if that makes sense. But at the same
No, it does, and I see the little seahorse.
yes,
in there.
and
Go
then
ahead.
finally, the last component, which is also very, very important, is that just above the mesh, you'll see a little PVC pipe. That pipe basically runs. It has drilled holes into it, and so our inflow is from that spray bar, and it sprays directly onto the mesh, so that no animal gets stuck.
Gotcha.
On it.
Gotcha. And real quick, I'm getting ahead of myself, I'm sure. But looking at this, you've got a certain age of seahorse here. We're going to backtrack because I showed the wrong picture first. But going to here, is there stages? Like at certain age, they're in here. At certain age, they're in here. At certain age, here.
Yeah.
Or, is that how it's working? Okay.
Yes. So.
Okay.
Yeah. So
go ahead.
So basically, of course, the chrysals are the starting point immediately after they're born. Usually, in my experience, seahorses are born within the first 3 hours. It's also fairly common to read that in the literature as well. Usually, the first 3 hours after lights on is when your seahorse will be born. Then we turn off the flow in the main tanks, which we'll show later as the photos come. We diligently collect them, usually sieve them out of the aquaria to prevent any pipetting, because the pipetting again applies pressure on a very gentle, on a very delicate animal. And then we put them into the chrysals. They stay in the chrysals for about 3 weeks, which is also coincidentally the most critical part of the seahorse life, or at least that is the bottleneck period that we found during my experiments. After the first 3 weeks in the chrysals, they will then move into the middle tier in the 20-gallon tank. Depending on, of course, on the size of the batch and things like that, sometimes we would move directly into the 29s. There really isn't much of a difference there. But anyway, week 3 to 5 is spent in the 20. And then 6 through 8, the 29s. And finally, after week 8 is when I place them into the 90. And then usually it was, again, it really depends on the batch size, but I would either split them immediately into the 290s, or I would hold them in the bottom in 90 and then move them to the top, or split them into the top later on.
So I have like about 500 questions about the system, but I'm gonna not ask them all just yet. But I do see tubs on the side. What are the tubs on the side?
Right. Just really quickly before we get into the tubs, the last thing I was going to mention about this grow-out setup,
yes, please,
is that this was the control setup. So this is what I would consider a very sterile, or at least an attempt at being a very, very sterile setup. Where there really isn't anything that is alive outside of the seahorse itself. Everything that you see in the photos, I don't know if you can tell, but there's plastic holdfasts. That's basically meshing tied to PVC But outside of that, there is absolutely nothing in those tanks. There's also very simple lights, nothing outside of like a T5 or fluorescent fixture.
Well, and that's said that I have 5 million questions. A lot of it goes back to your work with pathogens and why you chose to have such a sterile setup. But I don't want to interrupt, and I know I am again. So go ahead. Did you want to continue?
Oh, I was just gonna answer your next question, which was about the bins.
Okay. Yes.
The bins are good eye. There's 8 bins there. They're just your very simple 15-gallon bins from Canadian Tire. They were filled with Tisbe copepods, actually.
Gotcha.
So one of the, I guess, experimental variables that we were trying to also test at the same time was whether the lion seahorse specifically, because that's the species I did the majority of my work with, whether they would be better raised on copepods versus live brine, or a mixture of the two.
Gotcha.
So yeah.
Okay.
And did you have any results? Like, could you say that one is better than the other?
Not really. So I did not find a significant difference between feeding them purely live brine or a combination of both, or just Tisbe itself. And I believe that is and that is exactly what is in the literature as well. Line directus specifically is very well adapted to eating brine. It does not really need the copepods. Though I did find in the first few batches that the Tisbe do have a slight difference during the first week, and it's because the Tisbe copepod has a very wide size range, from about 50 microns to about 1 millimeter, and so what I was able to do was I think the copepods were allowing more of the batches to survive, even the very small seahorses that would normally die, and they still did later on. That would normally die were able to be carried through a little bit further, but overall there was no significant difference.
Were you enriching the Artemia?
Yes. So the brine has always been enriched with. We used to use 3 different supplements. The first one is Selco. That's the number 1. So it's just the essential fats. It's not only good for broodstock, just your regular fish, but also for especially for growing fish. We also used Algamac. Unfortunately, I don't believe that we used Algamac during these experiments, because the recipes ended up changing, and the new products are not very good, at least in my opinion. And then finally, we used VitaFish, which is just vitamins, basically.
Gotcha. So, going way off in left field, but I've got to ask, at which stage did you start? And I know we're going to go forward and back. Everyone who watches Wine Wednesday knows we do this. But I know there's more pictures to show. But at which in these stages did you start weaning them onto frozen foods?
Right. So that was actually one of the questions that that was, I think, in the comments a few weeks ago when I had joined the chat. I was hoping you'd ask. So we actually start weaning them off anywhere from about 4 to 8 weeks, and it's a little bit complicated. So during the trials, I started at week 4, but I did not actually find it necessary until about week 6 to 8, and it is week 8 to 10 that you really see a significant increase in growth, and that is because the majority of the animals are actually taking advantage of the frozen food between that period. It's usually 2 to 2.5 months, and that is when they're ready to transition.
So you weren't weaning them while they were in this system?
I was. Yeah. So like I said, go ahead.
I'm sorry, which tank were they in when they, like, because I'm just trying to figure out,
20.
Holly and I have discussed endlessly how hard it is to start weaning them because you're collecting uneaten food. So I'm just trying to see how that was handled in your process.
Yeah, so I started feeding them very, very minute amounts of frozen food as early as week 4. So they would have been halfway through the 20 gallons.
Okay, gotcha. All right, and let me back up and see. Did anybody else have a question? I can't see you guys while I'm doing
Well,
this.
we got lots that we'll save them for when the question period starts. If we can fit them in between your 500.
I know. Okay, I'll shut up. Let me go to the first picture here. Okay, that was that. Okay, let's go back to this. All right, what are we seeing in this picture?
Right. So I wanted to include these photos because, again, that was something that was mentioned before when I had joined in some of the other conversations. So these are our control seahorses at anywhere from about 3 to 5 weeks of age in the 20-gallon aquarium. So what you're seeing, and I'll try to be as detailed as possible, is you're seeing a close-up of that 20-gallon that was in the middle tier. You'll notice at the very, very back that the inflow spray bar is actually at the very bottom of the aquarium with various amounts of holes poked into it. So it serves as a nice spray bar. So again, even though we're out of the chrysals here, I'm still trying to drive that flow at the very bottom of the aquarium to prevent any settling, not only of the seahorses themselves, because they're freshly out of the chrysal where they don't really have to actually swim to be suspended in the water, but also at the same time to prevent as much of as much food from sitting on the bottom as possible.
Well, and that's because, yes, I am interrupting again, but that's because in the chrysal, they're tumbling, right? I mean, this is making them gonna, like, they don't, everything is in motion,
right.
Correct?
It's like turning one of our tubs on its side.
Yeah, exactly. Okay, and then what's in this picture?
And then if we go back, yeah. So this is the photo that I wanted to compare to. So if we go back to the other one, if you don't mind, just for a second, is now that you've seen the tank, look at the seahorses. So the seahorses are very, very, very dark. So they're pretty much jet black, and they're born this color. And they tend to stay that color until usually week 6 through 8, when they begin to really have a strong growth spurt and begin to differentiate male from female. So what I found was that in a very, very sterile setup, they will remain this dark color. Some of the questions that are very commonly asked is, well, I would like a yellow seahorse, or I would like an orange seahorse, or a red seahorse. So what you'll find in stores is that they're typically are fairly nicely colored. The problem is that that's not necessarily a really good judgment of health or what that coloration will remain into the future. So in a very sterile environment where you have black holdfast, they will mimic that coloration. So they exhibit phenotypic plasticity is what it's called, where basically they just blend as much as possible into their environment. So in the next photo you'll see is when they're raised in a much more colorful environment, such as when there's a brighter light, along with macroalgae and other things going on in the aquarium, they actually are coming out yellow. And this is at the exact same age as the ones before. It's just a zoomed in photo, so they look bigger, but they're the same age. So, anyways, what I find is that in juveniles in particular, especially with the rectus, coloration is not necessarily a good indication of health, or really anything at all. What you'll also see, and I think this is important for hobbyists, is when you go to the store, 99% of seahorses will be classified into this age group stage, is what I would call a juvenile. They're almost always sold before they reach sexual maturity. So they're just juveniles. They're maintained in bright light environments, and usually with what's very common is you'll see these nice plastic, colorful chain loops, and the seahorse is just mimicking the environment. So it does not mean that it's going to remain this color once it comes into your aquarium. It does not mean that it's healthy. It does not mean that it's any of those things that we look for typically.
Love it. Okay, I agree. And I have questions, but I'm waiting. What's this picture of?
I'll just say this. Maybe we can talk about it later as well. But I was just going to say, my view of coloration becomes a lot different when we're starting to talk about adult animals. And I did send a couple of videos. It's actually of Cuda, Hippocampus Cuda. It's not Erectus. But
do you want me to play them now, or do you want me to wait?
We can do it later. I was just okay. Foreshadowing, I guess. But coloration, I think, plays a pivotal role when we start to talk about adult animals. It really does speak. Color does speak to their mood, their environment, their overall health, and I'm seeing this from species to species as well. So I think it's fairly well applicable, not only to line seahorses, but to Cuda, red-eye, wide-eye, et cetera, et cetera.
Did you notice, I like I said, I have questions about the pathogen part of it, but did you notice a bigger success rate when you were raising animals in a really sterile environment, or when you were using macros, etc.?
So
I know.
No. It's a little bit of a loaded question, because there were pros and cons to both systems, basically. I think that I did not necessarily find that either system was perfect. It was also my first real iteration of this, and I think that with a lot more practice, that there could be a clear winner, both from, for example, a financial standpoint, from a biological standpoint, pathogens, et cetera, right? It's all labor. That's also very important. Labor is critical when we're talking about actually being able to run a business and things like that, making this sustainable.
Sure. Well, me and Holly fight all the time, because Holly's like, I don't want any of that crap. I want all artificial. And I'm like, I got to have my gorgonians and my macros and my snails. So when you figure out the answer, we need to know. Now I'm going to hush. And before we move forward into the adult seahorse questions and maybe talk more about coral and everything, I'm going to let Sophie take back over and see if there's other questions, and I will check the questions in the comment section. Go ahead, Soph and Ray, if you have your questions. I'm shutting up now.
Or you want me to ask now or later?
Yeah, I do have questions. So
you can ask now, Ray or Holly, whoever wants to ask.
Well, just to start off with, are you doing this alone, or do you have help?
I have a little bit of help, so but it was even at the time primarily alone. So yeah, labor was always an issue. So like I said, and I didn't really, I guess, answer the question. A sterile system tends to be overall a lot simpler and easier to manage. Yeah. So you could actually scale a lot higher and faster on a sterile system. But I think that from an ethical standpoint, the multi-trophic system is much better. The animals behave much more naturally. and utilize the entirety of the space. We don't really have to force them and nudge them into, for example, the spray bar being at the very bottom of the aquarium. They don't need that when there is abundant stimulation in the aquarium. So it's kind of like uh, uh, trying to enhance an environment in a zoo, for example, right? So it's and prevent the animals from getting bored. So the multi-trophic environment definitely stimulates them adequately. So that's, I think, partially why they also exhibit really nice coloration. Again, it's not tied to health, it's not tied to disease, um, but they do.
They're just happy. Hear that, Holly? I win.
And overall, I did find that seahorses are, at least in the Canadian market, they're a tough sell in the sense that I think if you were entirely, from a financial standpoint, if you're entirely relying on raising seahorses to sell, you probably will not be very successful overall. I think that the multi-trophic, yeah.
And that was one of my biggest questions, because we've had this debate multiple times, and I promise I'm going to be quiet now. But we've debated multiple times that for instance, Dan of Seahorse, when he was helping me figure out how to raise fry, it was all about the sterile, like he had these huge 90-gallon tubs in a big, environment. And as a hobbyist, that was very difficult to do. And I always had the question, if you're raising these seahorses in a very sterile environment and then selling them to a tank that is not so sterile and is not going to be so sterile, is that really the best way? Or would it be better to introduce them to things, like a kid scraping his knee and then not getting something terrible down the road? I don't know. That's a deeper question, I know. But just your brief thoughts on that, and then I'll.
Yeah, it's always something to think about. I think when you're breeding these animals, I did actually find that overall, sterile environments, so having to use plastic is more abrasive to seahorses than macroalgae is. I'm not a huge fan of live rock in seahorse aquariums, but it is something that is commonly used. But does that? It's always very difficult to follow the animal through. I understand your question. It's
There isn't an answer. I know there really isn't. I'm giving you questions that you can't answer. I really do understand that, but it's like the theme of the hobby. It's like, do you go with these sterile tubs, or do you try for more green water kind of thing? And there is no answer. So we're gonna need you to step it up and get us some answers. Okay, all right. No,
Okay, can I ask some more here?
Okay, I'll shut up.
Why did you choose for aquariums rather than tubs? The tubs, you can get the circular motion going in there and keep them from getting at the top there. And I found it much cheaper buying tubs and setting them up, because basically the plumbing wasn't costing me any more for tubs, but the tubs were so much cheaper than buying tanks.
But how big were your tubs, Ray?
I'm sorry?
Sorry, I said how big were your tubs? Like, his system looks small on a picture, but it's.
I had the smallest one I had, I think, was 17 gallons, and I didn't have any as big as Dan, but my biggest ones I think were 38. I think they were 38 gallons, but I like the fact that you can keep them in motion, and it was quite easy to up the flow a little bit if you found that they were sort of getting at the top, staying at the top more. and it avoids the fact of having to form a creasel piece to put into your tank. I'm just wondering, did you consider the tubs, and did you choose the tanks for a specific reason?
Yeah, that's a great question, and it's something that that Zach and I are constantly thinking about, and I think it's a very applicable question across every species that is being bred in captivity right now. It's not just seahorses; it's every fish, it's every invert, and coral even is where it extends to. You can always get fiberglass totes to raise coral. You don't necessarily need aquariums, and it's a very simple, shallow answer. I think I would say is that it's just nicer from a visual perspective, and to have aquariums, and which is why we. But now I will tell you is that we do use some bins and some aquariums. So because we're very short staff wise, and labor is always something that we have to play catch up with. Having the aquariums there versus the tubs, it kind of stimulates us visually to, hey, I need to attend to this right now. It's always, for example, I'll go to coral really quickly and say that for a brief while we raised coral on multiple tiers. And I will tell you right now that we never did the statistics on it, but the bottom tanks always suffered way more than the middle ones that were eye level, or the ones on the top tier. So it's basically just simply for that. It also, to defend a thesis, whether it be a master's or a PhD thesis, photos are very important. I did not include a bunch of those, but I also had to take measurements of seahorses, and the way I did measurements is I would stick a ruler in the tanks, and then I would take photos with the camera, and that to me was the least invasive method in doing so. And then from there, I would use a program, just a simple image J, to analyze each photo, set a scale, of course, based on the ruler, and then take average measurements, and then I would track the animals from 0 days all the way to about 12 weeks of age. And so that
I love that honest answer. Oh, sorry, go ahead.
No, I was just going to finish off that would have been an extreme challenge with trying to do it in tubs, and trying to get those animals nice and level, so you can get the tail all the way to the snout. But I completely agree with you. I think that the tubs are a fantastic option. They're inexpensive. There's lots of benefits to them. Yeah.
Well, I love your honest answer. And the thing is, though, with the tubs, what I came across is you can only keep them in those tubs for so long. I did have little ones. I didn't have the massive ones that Dan had. But still, once you start weaning onto frozen stuff, it's so much easier in tanks because you can see, what's going on. But, all right, go ahead, Ray.
I just have a question on UV and one on copepods. Now, with the copepods, the only one I found that I really needed the copepods for was pelagic fry.
The which, sorry?
The only ones were the pelagic fry that I needed the copepods for, because they were smaller, like the reed eye. Well, and you mentioned CUDA. Well, it depends on which CUDA, because the CUDA complex has been kind of buttered over the years with so many offshore companies. Found it was easier to ship seahorses and call them CUDA to get them across customs, when there might have been something entirely different.
Yes.
But anything like the reed eye that have pelagic fry that don't hitch at birth, they're smaller, and I found that it was very difficult, if not impossible, to even use the smaller Artemia from San Francisco. Those were a little bit smaller, and they would take to them a little bit sooner, but I still had to have something smaller. And when I didn't have copepods, I was using the
Rotifers?
rotifers, yeah. And people told me it wouldn't work. Well, I raised an awful lot of seahorses, starting them off with the rotifers. And well, back in that era, we didn't have the Algamac 3050. We only had the Algamac 2000 back then. And but I didn't really see any difference when they went to the 3050. So I'm not sure what improvement was made, but it didn't make any improvement in the results in raising the seahorses, not from my perspective anyway. But and the other thing was UV. Almost all, I've never come across any seahorse disease yet that was caused by a pelagic bacteria. They're all benthic bacteria that cause it. And so I'm wondering why you use the UV. I think Dan used it, and I think some other people have used it. But I'm curious why you chose to use the UV when it's not going to affect any benthic bacteria, which are the biggest problem we have.
Right. So going back just to the copepods just for a second. So the Tisbe specifically are. I did want to use them and to test them on the CUDA because I had. So the literature states that it is indeed REDI and CUDA that require them. Usually during the first at least couple of weeks. The Tisbe copepod as well, for those that don't know, has the highest percentage of polyunsaturated fatty acid content per body weight, and they do not actually require any supplementation at all, and they still rival supplemented live brine. So between that and their wide range of body size, they are quite. They were my number one choice for trying to see if we can find a difference. We also did try rotifers as well. This was some preliminary trials that we did, and we did not find any significant difference between that and live brine, at least with the rectus. I think that the rotifers, unfortunately, are just a little too small. Maybe the first week, I think, is beneficial, but the labor associated with raising rotifers, I think, is quickly negated by
Yeah, I only used the rotifers until they got on week to 10 days, and then they could take the San Fran Artemia, and then a week or two after that, they switched them over to the Great Lakes.
Yeah, and then your second question about the UV sterilizers.
Yeah.
So we almost instinctively run UVs on everything that we have. So we run it on all of our fish species, all of our inverts, all of our corals, and it's again it's a fairly simplistic question. So you are correct. A lot or most of the pathogens that affect seahorses are benthic. But at the same time, we are constantly stirring things up as well. So to some extent, that there is some benefit to running the water through a UV sterilizer, and then there are some indirect benefits as well that are not necessarily pathogen related, but rather nuisance. So a lot of nuisance algae, cyanobacteria, dinoflagellates. Things that can creep up in a very high productivity, high nutrition environment, and that would cause a lot of extra work for us, basically.
Yeah. Okay, I'll turn it back to Sophie.
Okay, and so Bella's seahorse beauties went straight to how many seahorses, with all this fancy setup, the research, how many seahorses approximately, I don't know if you know the exact numbers, but how many seahorses have you raised and sold?
We probably I think so. We've been raising seahorses for quite some time now. We've probably raised certainly into the thousands. How many thousands? I have no idea. It's I've personally raised only 3 species, which is Erectus, Cuda, and Wide-eye from Australia. But I have had in captivity Reed-eye as well, and then and then Zach's mom actually has some. Oh, and we also had a hybrid species as well, which was something Ray touched on when talking about the Cuda. Most typically, you'll find Cuda that are actually hybridized either with Reed-eye or Erectus, and they're coming from typically from Asia. They're marketed extremely well. Unfortunately, they're to some extent sterile, so they usually will produce fry on the first batch, but those fry will either die almost instantly within the first week or so, or they just do not grow at all.
So just for your, oh my gosh, where am I? All right, I'm going to turn that back off because I'm green and and pink. Okay, so
what kind of wine are you drinking?
Right. That is the wine, y'all. I don't know what's going on with my camera, but what I was going to say is that's really interesting. As you research further, the information that I've gotten, I did have hybrids also, and they never bred for me. But I've heard that many people have had success if a hybrid breeds with a, I don't know how to call it, pureborn. So like if a hybrid breeds with an Erectus or a hybrid breeds with a Reed-eye, then you have more success. Just a thought for you to look at, in the future. Holly or Sophie, go ahead.
Well, I was just gonna let Holly ask her question now, because I know you have one.
So I think I remember you saying when we were looking at the picture of your rack system set up that you just had one sump, right?
Yes.
A 20-gallon for all those tanks?
Yes.
So I'm wondering what kind of water change schedule were you using? Like about how much water were you changing out, and what did your schedule look like for that?
Yeah, so that was the other difference. I'm not sure if I included a photo of. I hope I did anyway. A photo of the multi-trophic system. So it's essentially a mirrored setup of the sterile one, but it was a lot busier. Anyway, I'll answer your question. So the control system was siphoned the 2 chrysals on a daily basis, sometimes twice a day. The 20-gallon every day. The 29s usually every other day, and then the 90s every other day to every third day, and then the volume of water was it was a ton. I mean, we would go through instant ocean bags on a daily basis, so it was hundreds and hundreds of gallons a week.
So would you say then you're changing out like 100% of the water about every 2 days or so?
I probably wouldn't say every 2 days or so, but certainly I would say that maybe 1 to 2 times every week. I think of full system volume. Yeah.
Okay, and I wanted to ask about the salt, because I know I've asked this question like 2 weeks ago, something like that, because I wanted to switch salts. And I know that you raise, like, you have corals, you have whatever, like more than just seahorses. Currently, I'm using the red sea salt, but I would be worried that if I would change, it would be to instant ocean, because that one's probably cheaper, that it would affect the seahorses or like the corals in the tank negatively.
No.
No.
I wouldn't be concerned about it. So there's, I will say that it's, at least in Canada and a lot of the Canadian groups, there is a lot of discussion at the moment regarding salts, and that instant ocean has somewhat of a bad rap lately. But yes, I only use instant ocean. I actually even downgraded from reef crystals to just pure instant ocean because of the sheer volume that we're using now. So yes, it is a monetary thing, but at the same time, I find that um, for fish breeding and raising and for inverts, the uh, the salt brand does not matter at all. I think that instant ocean is more than satisfactory for any fish, fish only tanks, fish breeding, any inverts, etc., etc. It has more than enough calcium in the water and good enough, more than good enough alkalinity level that no species would be of concern. Um,
Totally agree.
yeah, I do not.
I've never used anything else, and it's always been fine for
And
me.
it is something that Instant Ocean brags about in every single possible opportunity. Is that while they supply practically every North American large aquarium and with their salt. So um, they are certainly they're also one of the oldest salt brands that exists. So I do not find them to be an issue when it comes to coral. I'll even say that is that where coral culture and coral aquaculture and the hobby and everything else, we're already moving so fast towards basically supplementing every little element into the aquariums themselves that the salt, I think, is just the very starting step. And it's not necessary for the salt to be immaculate, I guess, in its mix to provide a good enough base for us to build on.
Okay. And Ray, I think you
Just
had a question.
to just to make you feel a little bit easier on the salt you're using, back in the late 1990s, I dug up a formula by Frank Malero on chemical oceanography, and he had a formula there for making up your own salt water. Well, instead of doing the full formula, I took the top four salts that were used in it, and they were the major ones. The rest of them were all kind of minor. And my thinking was that these four salts would have a lot of impurities in there, which were supplying a lot of the minor ones. And I ran it by Randy Holmes Farley, who was a scientist, and he felt that there was no problem with that at all. And I ended up starting off, I only had reef tanks back then. And so I started off with using a little bit of that mixed with Instant Ocean. And I gradually started using it up to 50%. So 50% of it was my homemade salt and 50% was Instant Ocean. And it was purely because of cost, because with 1,000 gallons of water in the basement there, that it was a lot to have. Even though I wasn't pinched for money, I didn't like spending that much on it. So when I could do this and I could buy these salts in bulk, well, one of it, the easiest one was buying salt or the water softener.
Yes.
So, and if you buy a pure water softener salt, that's the biggest salt that goes into it, and that's the cheapest salt you can get. But basically, I was mixing up salt. I'd be buying in bulk and mixing it up, and it was less than half the cost of buying IO. And IO was always the cheapest.
Yeah.
Yeah. Well,
so I've used IO for over 30 years.
I think making my own salt is a step right now that I do not want to do.
You're not using enough of it to make it pay.
Yeah.
You can't go out, I can't even think of the name of the store here in London now. But the factory where I was getting the salts from, you can't just go out and buy like 20 kilograms or something like that. You'd have to buy larger volumes. And so for me, it was practical because of the amount that I was using. But basically, I brought it up just to give you, to let you know that the salt doesn't have to be perfect to be successful with the, well, I found no problem with the corals either, but especially when I got into the seahorses. When I first got into the seahorses back there in 2002, I actually didn't, I used straight instant ocean because I didn't know at that time how sensitive seahorses would be. And it was something that it took me, I think, a year or two before I got up to using 50% on seahorses. Just feeling it out, because we didn't know the same kind of things about seahorses back then that we know now.
Yeah.
But you won't have any problem.
So I'll probably change to it. Then
if you're worried about it, you can mix half and half with the one you're using first and carry it over like that. Like when I'm changing my dogs over on their food.
I'll do it like that. All right, and then Bella's Seahorse Beauties asked, do you have heaters in your tanks?
No. So at all costs, we attempt to avoid any heaters in any tanks. So the heaters basically remain in the sump area, but outside of that, when we're breeding any type of animal, I recommend that you leave the heaters out in all possible scenarios. So you can use any type of water bath if it's not a plumbed recirculation system. Otherwise, if it is, then I would leave the heaters in there.
Okay, and then also,
so what temp is your system, real quick? What temp do you run at?
So we run seahorses anywhere from about 76 to 78 degrees Fahrenheit, or about 25 Celsius. But you can certainly do them cooler.
Well, I do mine cooler to prevent bacteria growth, but I think it's important for people to remember that you're doing huge water changes too, so you're keeping down the bacteria growth that way. Sounds like.
Plus, it's keeping fairly sterile systems.
Yeah, exactly.
I was just going to say the temperature is also directly correlated with usually with the rate of growth. So because it dictates the metabolism of the animals. So cooler temperatures, just the same as bacteria. If you're raising them, at 70 to 73, 74, that they will also grow slower as well. Not dramatically, but over the course of years of raising animals, you will definitely see some differences versus raising them at 78 or so.
Interesting. Good to know.
Well, sure. And we're going to ask you more about coral and inverts and other fish. But like with seahorses, we absolutely recommend that any newcomer trying to keep seahorses goes with a lower temperature, because you're absolutely right, they'll grow faster. But for a newcomer that doesn't have that sterile system, it's going to be much more beneficial for them to be at a lower temperature, because then about the pathogens, all about them, right? So it ends up a better situation if you have a smaller tank and a smaller system, and not that many whatever to go at a lower temp. But okay,
all right. And then
and Ray said,
do you guys want to go into the direction of like inverts, talking about inverts and corals? So do you raise any other inverts?
I do. So I actually think I got confused earlier. So I do raise Bergia, but I was actually talking about peppermint shrimp earlier. So those are two key inverts that I'm working with at the moment. They're both
absolutely essential to seahorse tanks. Love them, and I'm sorry that I confused you. That was my fault. But go ahead.
Yeah. So those are probably the two that we currently raise a lot of. So the Bergia is, for those that know, it's basically it's a sea slug, a nudibranch that only gets to about an inch in size, and it feeds exclusively on Aptasia. So they're fairly easy to raise, and I think that pretty much anybody that would be interested in trying to breed something, I think the Bergia is a great opportunity. They have a very short larval life stage of only about 2 weeks, during which time you don't really see them in the water, but that's okay. As long as Aptasia is present, they'll be fine. And you will probably be able to raise a few quite easily in a very simple 5-gallon aquarium with an air stone. They do become a lot more challenging trying to scale them into raising thousands at a time, and that is actually because of the Aptasia itself, and the Aptasia actually becomes your best friend. It's the Aptasia you're trying to raise, not so much the Bergia. It becomes flip flop there.
Which is insane, and you need to stop giving away your secrets, dude. You're
That's
trying
okay.
to sell these
No,
things.
that's
But no, I'm teasing. I love people that come on and want to share the knowledge and and help everyone. And I do absolutely want to know more about peppermint shrimp, because my peppermint shrimp gave birth all the time, but I had no idea. But go on.
no,
I'm
no,
sorry. I didn't mean to
it's all good.
interrupt
So
again. Go ahead.
yeah, so peppermint shrimp. There's usually, at least in Canada, we get primarily Lismata bogesii is or however you pronounce it. That's typically the species that comes into Canada. It's a fairly dull red or burgundy shrimp with a blackish tail. And the black can fade in and out quite easily depending on on the shrimp's mood. Unfortunately, that is not the correct species of shrimp that you should be buying and putting into your aquariums. They are actually they're pretty good at eating coral, particularly LPS and softies, given the opportunity and being in larger numbers, especially. But anyway, I raise, like I said, Verdemani, which is a shrimp that looks a lot like camel shrimp. So it's a clear shrimp body with bold red lines, and they're found in the exact same region of, like I said, Florida and the Caribbean. So that's why they're usually easily mistaken for one another. But for whatever reason, Verdemani does not actually come up to Canada. It's just a species that comes up through Biota, actually, the company in the U.S. that breeds fish and inverts as well.
And then Kelly was asking, do any of the seahorses eat the shrimp?
No, the seahorses, the seahorses and the shrimp coexist very well, work very well together. So the shrimp, not only do they eat Aptasia, but they're excellent detritivores. So in general, the purpose in them during my experiments was actually to serve as a detritivore. So to limit the amount of labor that I had to do by eating leftover food in the aquarium. And they do that job very well. They're not picky about what's on the menu for dinner, and they grow well. They as long as they're fed well, you're able to maintain them in fairly large numbers as well, so into the hundreds in a fairly small aquarium. But they do turn cannibalistic if you don't feed them. Yeah, so the shrimp itself has to be raised separately. So as Ray mentioned before, they're excellent for black bins and things like that, tubs. You don't have to have very high end equipment at all, but they do have a fairly lengthy larval period. Of about anywhere from about a month to a month and a half, even almost 2 months, depending on temperature and food and things like that. and usually it's larval period that length are challenging for most hobbyists to get through, because during the whole time you have to feed them live food, whether that would be pods, rotifers, brine, etc., etc. It's a lot of daily labor that you would have to invest to raise something that has a long larval period like that. And then from there, once you get them through the larval stage, they're easy. There's no problem. You can integrate them in pretty much every aquarium. They're good in a 5, 10, 15, 20 gallon tank, and et cetera, et cetera.
So it's just the larval period, but they're not like mysids that eat the young and all that, right? Like they're
No. But you do separate. So you do have to separate, of course, the adults from the larva. Um, is yeah. And I don't find them to be cannibalistic at all until they complete metamorphosis, which is the transition from a larva to an actual animal, or an actual shrimp rather.
but it's literally like having another fry tank
Yes.
that you have that you have to feed, and live foods and keep it clean and all that good stuff. So it might not be worth it for the average hobbyist, but for anyone that's looking to go commercial, you just gave them a lot of good information. Okay,
And
go on,
then
Sophie.
I think what a lot of us are wondering, since you're talking about all these things that you grow, where did you buy them from? And for all of the Americans here, can you ship to the US, or how far out do you ship?
So we ship in all of Canada. So from BC all the way to Newfoundland. We do not unfortunately sell anything to the US. I'm
You're fired. No, I'm just kidding. I'm totally
You pay
kidding.
for the CITES then.
Yes. So it's now going from across the border into the U.S., for example. It's you have to have certain permits, and they are and there's a lot of restrictions, and then you have to pass various tests and things like that to ensure that you are able to move animals across the border.
Is that
Seahorses in
something that you're thinking of doing in the future to be able to ship to the US, or you don't want to get into that?
it would be great. So my only goal really is to is like I said, to continue to withhold or to continue to hold my ethics and morals, and to continue to attempt to curb wild harvesting as much as possible, and to provide quality animals for the aquarium hobby, and to provide more than anything education. I think that's really where I think that my experience would be more valuable long term to people in people in the U.S. and things like that. I think there's plenty of sources in the U.S. itself that you would be able to find.
Nope. There's not. We need you. If you need help figuring out shipping, I'm here to help you. And I love that you want to educate. And I love that what you just said. That's going to be the next post, because that's amazing. And I believe you. And I think you're just trying to help people and do the right thing. But we need you. So there's no more sources here. And, we need you to get on it. So, but okay. Backing up now again. Go on. I didn't mean to interrupt. Go ahead.
Yeah, so I was just going to say that with seahorses specifically, because it's species dependent, right? So depending on what we're talking about, and this would be a lot more Zach a savvy question, and then it would be me type of question. I'm very much focused and tunnel visioning typically on the nitty gritty of breeding species, and trying to figure out new and creative ways to slow down Zach's progress, and to, hey, I think we should build this or we should build that, but leaving a lot of that tinkering to him, in terms of the setups themselves. But anyway, seahorses specifically, they require CITES. As far as I'm aware, There is very, very limited, if any, wild harvesting at all of seahorses of any species. So basically, everything that's coming in from overseas or across borders, things like that, are captive bred animals. So yes, exactly. They, as Ray said, they require CITES. So even captive animals, and captive bred animals tend to have a lot easier of a time crossing borders than wild animals. So yeah.
Well, I do remember Dan saying that. I mean, we understand that it's a pain in the butt because they want to know, like, where did you collect the seahorse? We didn't collect the seahorse. We bred the seahorse and raised the seahorse. Well, then there's a problem. I mean, like, there's all this crap that you have to go through, red tape. But Sophie's going to ask you then Ginger's question, because we need you. We need you in
So
the U.S.
maybe the
But go
solution,
ahead.
though, would be to have a partner in the U.S. So we have our own supply here, but we can consult back and forth. People can travel.
Whoops.
Dimo could come to the United States and help somebody here set up their own, and vice versa. We could
Look,
come and
Dimo's
look at
like, listen,
Dimo and
y'all,
say, "Hey,
I
how are
just
you
came on
doing?
here to
What's
educate.
your setup?"
God dang it. Sorry, Dimo.
No,
we want
I was
more.
just going to say,
And we want, and I do want to say too, we need to get in touch with Zach, your partner, and get him in here too, because we got questions. But okay, let Sophie take
I was
over.
just going to say, you guys are alluding to a good point, which is the biggest issue I think that seahorses in captivity are suffering from is that we're creating these very isolated populations of animals. We have very, very, very poor gene flow, is what it's called, or genetic diversity is more layman terms. So we have populations that exist in various countries that have been bred for 10, 20, 30 years. They migrate from aquarium to aquarium, but again, they're usually maintained within a fairly close perimeter. And those animals are never exposed to different gene pools. And unfortunately, what it does in seahorses is that it definitely affects fecundity, which is basically the size of offspring being born, the overall batch size of being born, and I've actually seen deformities, whether that would be missing tails. I've seen or partially missing tails. I've seen partially missing snouts. All sorts of different deformities right from birth, and that's the problem.
So that's why we saw in the U.S. We had. I'm sorry, you guys. I can't turn on my camera. When I do, I turn into this. I'm sorry, but I am here. And I was going to say, we had an experience where a bunch of people many years ago, seahorse breeders and hobbyist breeders and hobbyist enthusiasts, they got fuscus. Fuscus. I might be saying it wrong. Fuscus. I don't know. Anyways, a species of seahorse, and we got them. They bred them down, and everybody was successful for a minute, and then eventually, there were no more, because you can't keep breeding in the line. So, but I think the key is, I think the sustainable action is to have respectable breeders like yourself, scientists, people that know what they're doing, deal with people like Chris Carey, who have the ability to collect new blood, and very few, to where it does not affect the ocean. But then, they work with you, people like you, to do breeding programs so that there's new blood in the system, but we're not affecting the wild, and we're still providing healthy seahorses for captive environment, is my thoughts.
Yeah, and
what are your
I would
thoughts?
say, because I know, Dimo, you said that you provide some seahorse. I might be completely wrong, but last time when I got my seahorses, you said that you provide the seahorses for candy corals, right? In Toronto?
Yes.
Yeah, see, because I wanted to get, if I were to ever kind of bring new seahorses in, they would be from kind of the same line that I have. So then it just becomes like a problem. But then going back to the shipping, because I think all of the Americans are not satisfied with the businesses they have in America. Zen is asking if someone had an import license, could you sell to a U.S. person, or do you have to have the license on your side?
I think that so both parties have to have usually a broker that is on either side of the border that mediates the transaction that would occur. That is at least to my knowledge the appropriate manner, and usually in moving animals across borders like that.
Okay. And sorry, we're jumping back and forth. But going back to the diversity, have you done anything? Have you shipped imported seahorses in to diversify your bloodline, I guess?
No, no, and I'm essentially alluding to it's. I have the same problem as what I think everybody does, is that our Erectus line should require the addition of new animals. But I'll be honest, I have a hard time finding somebody that regardless of where they are to reliably say that they have, first of all, a purebred Erectus line, and secondly that they could supply. So our seahorses actually originally are from Ripley's aquarium. So we have as a laboratory, we had a very, very good relationship with the people there. In fact, my professor's probably star pupil. She's the veterinarian currently of the Canadian Ripley's Aquarium of Canada in Toronto. So anyway, that's where I've never imported seahorses. But I do have some friends, I guess, in the industry that do, and that is how we acquired our Cuda. So they came from Australia and the Wide End.
Well, I was just gonna say, please get in touch with me on the side, because I'm nobody, but I do know people, and
And that's
I
very
can
important.
put you in contact with Chris. Yeah, with Steve McLeod in Australia, with Chris Carey, Chris Carey, with if we figure out the U.S. Canada thing, Paula runs Dallas Aquarium. I mean, I want to help you because I see what you're doing is making a difference and you're doing good things. And so, just keep in touch with me on the side. I can absolutely put you in contact with these people and, get you sustainable, good, healthy new seahorses, new blood.
Wonderful. Thank you.
But we did want to ask too about, I think Sophie's going to ask you, and let me find the comment. Yeah, let's ask about this real quick.
Okay, so what type of different coral species do you sell? And I know you sell a lot, but an overview of your coral.
So just quickly about coral, we have I think at least a few hundred different species of coral. So everything from soft coral like zoanthids, leathers, things like that, to LPS and SPS corals. So basically, we have, I think in essence, we have pretty much every single species that is. I think species is a little too much, but we have some of every type of coral that is able to be propagated in captivity. So basically, where we lack, I guess, compared to your typical retailer or a wholesaler or whatever, is we do not have any of the very popular scholemias, gantophilia, lobophilia, things like that. All of the very large polyp LPS corals, because they just do not tolerate fragmentation in captivity at all.
Okay, and then so as someone who likes the natural tanks, I know that you've kept your seahorses with macroalgae, but do you keep them with any corals ever, or have you done any experimentation with that?
To be honest, not really. Zach's mom, who really maintains more display seahorse aquaria, does keep them with some corals. Particularly, I think gorgonians are your number one coral that you would mix with seahorses. But I would say that practically every soft coral, I think, is a good candidate for a seahorse aquarium. Um, what else? I would definitely avoid any SPS for sure. And the reasoning is not so much that the seahorse and the coral cannot coexist, but rather that the SPS, or small polyp stony corals, things like Montipora, Acropora, things like that, they require high flow rates, and it's that would cause the seahorse to feel uncomfortable in that environment.
Okay, and the flow is another thing, because I know I've talked to you and you say they require low flow, but then the general, like, there are a lot of people in this community that say, and like I know Ray, for example, had a lot of power heads in his aquarium. So what are your opinions on like flow?
Okay, so I think that flow in general, high, medium, and low, is kind of a very broad general statement. So I've never had power heads or wave makers in any of my seahorse aquaria, but we have always had oversized return pumps. So because we always run on recirculation systems, um, yeah, they're typically pushing anywhere from 10, 15, 20,000 liters per hour, so to large pumps. Um, yeah, so I think that well placed flow is more important than just general high flow. Does that make sense? So,
Yeah.
for example, the utilization of spray bars and using them in, creatively to because it's really, I don't believe that the flow is really that high, or that has a direct impact on seahorse health. I think it has a lot of indirect impact on seahorse health, and that is when we're
Oh, wait, hold up. Totally disagree. What do you mean?
on.
How,
Okay, so
what do you mean by indirect versus direct? Meaning my seahorses flow dance, and I had a tank that was like failing miserably, and I added flow, and all of a sudden they were happy.
okay.
So what do you mean?
Okay, so what I mean is that, for example, with corals, gas, exchange is so between the host or the animal and the environment is facilitated through flow, meaning that without flow, a coral, in a lot of ways, a sea-style animal, will not be able to perform adequate nutrient intake or uh, what's it called, and waste expulsion. Does that make sense? Because a coral, in its essence, really has a mucus layer, and it is.
It needs the flow
Right.
that's direct. So you're saying that the seahorse only could possibly care about the flow because it likes it, or it doesn't need it, but it
I think
likes it.
yeah.
And it can
What I'm
move
trying
out
to
of
say,
the way.
what I'm trying to say is that it's not necessarily a vital component to its
Yes. Okay.
existence, and that's why I'm classifying it more into the indirect category, because I think that
I
you can
understand.
have seahorses, yeah, in various degrees of flow, and all of those will still be just fine. I think that like I said, flow is important to, in a sterile system to prevent them from detritus from settling constantly. I think flow is also very good. I think is what you were trying to say, as in it stimulates more natural behavior. I think it stimulates them to move, to engage more with their environment. I think it makes them more active in general, that enhances not only behavior but feeding, et cetera, et cetera.
Well, and for us lazy hobbyists, that have macros or coral or whatever in the tank, the flow helps us not miss uneaten food and things like that. It does depend on whether you're talking about a very sterile tank where you can see everything and you have a system that you don't need that higher flow. But in a hobbyist tank like Zen Ginger, I'm calling out to you, Mama. If you're going to have other things in the tank, then having that higher flow rate helps make sure that nothing gets stuck underneath something that you can't see or whatever and becomes, additional organic waste. But no, I understand what you're saying now. I thank you for
Not a
explaining.
problem.
Yeah, and I'm just gonna add
And
that I did for a while keep the flow lower. So my power head, I put it like all the way to the lowest. It was barely float, like there was barely any water coming out of it. And I did notice a buildup of detritus. And Dimo, I don't know if you remember, it was a while ago. I sent you some pictures of my gorgonian, which was dying. That gorgonias has since been stolen by my pistol shrimp, this whole thing. But when I did increase the flow, the detritus kind of started lifting, going into the filter. I got it out, and the gorgonian actually did a lot better.
Yeah. It's
yeah, no.
Kelly, I don't remember. Was the question answered? What is a recirculation system?
I'm sorry. Oh yeah, what is a recirculation system?
so
What is
it's
that?
a recirculation system. So it's just a closed loop, basically created by PVC or other type of plumbing that circulates water from one aquarium into the next, and particularly from the picture that I had at the very beginning, from the sump to all of the other aquariums, and then from the other aquariums it overflows back into the sump. So that's what you would create a full loop, right? Versus, for example, most, and I'm going to say most, I think things are changing, but in the scientific world and in commercial aquaculture, most breeding facilities are either on the water or very close to the ocean. And so I would not consider those a recirculating systems. They really are, to a certain extent, just pushing water from the ocean into their setups and then pushing the waste back out. So I wouldn't say that that's a recirculation system, but that is changing, and the reasoning behind it is both to protect our natural environments and to also protect our captive breeding programs because
So can you talk for a second about what's wrong with pulling water from the ocean and pushing it back out? Because I totally agree that's not the same.
it goes both ways. So it's bad for broodstock and for breeding because the ocean, while it's very very clean and natural and perfect, it has a ton of opportunistic pathogens that, given the
Yes.
very, in comparison, small environments, very compact environments, and very productive environments that we as humans build to raise various species, can just absolutely explode and wreak havoc through our systems that would not normally do so in the ocean. So that's the first, I guess, lesser impact that they have, and then the second impact is, of course, we cause, so in any biology program that you enter in Canada, I can pretty much generalize this across the board, is that you will find a class that will talk about eutrophication, or how we're constantly polluting the water, and the pollutants primarily, and when we talk about aquaculture, is nitrate and phosphate. So we're constantly dumping excess nutrients into our natural bodies of water, and then that leads to a process of eutrophication, which essentially you starve out the environment, or you supersaturate it so much that you have bacteria explode, it consumes all of the oxygen, and then you're left with absolutely nothing.
Great explanation. No, thank you. Me and Sophie were talking earlier about, you guys are using all these words, but you just did a great explanation of what you're describing. I really appreciate that. Sophie, did you have something else to ask about that?
No, not about that. Are there any other questions about that in particular?
Yeah, I think this one is one we missed, and then there's a couple others.
So Zen was asking, are corals treated like other animals regarding sales and shipping, or are they special like some things are?
So I will say this: I think that every animal is very, very special. So I also get asked a lot, what's my favorite fish or coral or invert? Every single one of them, every single one that I had the pleasure to work with is my favorite.
Well, again, you're fired. It's supposed to be seahorses. No, go ahead. Go ahead.
I will actually admit that during some parts of my thesis, the seahorses were actually some of my least favorite animals. But
I know, right.
They're needy.
Yeah. Yeah, especially when you're trying to write a thesis at the same time, and yeah, it's fun. God. But anyways,
I think she meant like as far as shipping to the U.S.
Yes.
I think that was her.
So again, with corals, the differentiation there is that soft coral, so any coral that does not have a skeleton, is rather easily migrated between the U.S. and Canada, and does not require CITES permits. But anything that does, so any LPS or SPS coral, is strictly controlled across the borders, and it does require a CITES permit.
Okay. Interesting.
Well, and I wanted to add too, when you guys were talking earlier about why not to maybe keep SPS in a seahorse tank, it's also because, the seahorse will hurt the coral, right? The seahorse will hitch on an SPS, and then all its polyps get damaged. And then, also the SPS needs a higher temperature. And as a hobbyist with lots of things in your tank, you want a lower temperature. So it just doesn't really jive. I've seen people with 300-gallon tanks pull it off. Absolutely. But it's more about water volume, size, things like that. All right. I'm done jammering. What else, Sophie? What do we got left?
We don't have other questions, really. This is off topic of what we're currently talking about, but another thing I wanted to bring up tonight is that, so this was a while ago, my female seahorse had parasites, right? So we were talking about like the itching, that were signs of this. And I remember emailing you, and I remember talking to you a little bit about it. I did end up doing the freshwater dip. That was before you told me what you did. And I remember that your opinion on freshwater dips was actually against it. And that's been something I've been kind of thinking over. And yeah, I wanted to bring up while everyone is here.
Yeah, great. Go ahead. Why are you against freshwater dips? And, this isn't gang up, because I have certain opinions about freshwater dips that are negative also. So what do you think?
Yeah, so the it's not so much that I'm against freshwater dips, but rather I am trying to. So we were talking about ciliates, right? So that was the cause of concern. So ciliates are, as I tried to explain, ubiquitous in pretty much every environment, and
Yes.
that's where really my problem with freshwater baths comes from, is the fact that and you did mention this as well, is that it's very temporary relief, right? So it's because it's isolated directly to what is found on the animal itself, and I begin to weigh the benefit of ridding just what would we call that, 0.01% of the population, and trying to weigh that against, because I think that everything in this hobby, and particularly the more complex a system you begin to create, I think everything becomes kind of a weight scale, and you have to kind of weigh the your actions and the benefit of those actions, right? So freshwater bath, I think, is great. It's fairly uh, yeah, a seahorse will tolerate that fairly well. But how much good is that going to do? In 10 minutes after it's placed back into its environment, an hour, 2 days,
Mm-hmm.
5 days, 7 days, right? So, and then I begin to also think about how much stress am I causing my already stressed out animal,
Yes.
and then I begin to worry all too much, from way too much personal experience, of particularly gas bubble disease. That is my, at least in captivity, one of the number one issues outside of bacterial and parasitic infections is gas bubble disease. And gas bubble is
Mm-hmm.
caused predominant. Well, it's not predominant. Yeah. So if it's if we're calling gas bubble disease a cause of disease, because it typically is just a symptom of an underlying condition as well.
Yes.
But if we're calling it as the predominant cause of concern, then it's all caused by us. Really, it's a captive environment, and
So what is your answer to someone that sees a seahorse with either gas bubble, tails on the bubble, or, a particular seahorse is bloating up and has internal gas bubble disease, or you just see them scratching and it's only a couple of them. Like, it's not all of them. Most of them are fine. Only one or two are scratching, or your fryer scratching. And that was like a 3-part loaded ass
no,
question. Sorry.
it's okay.
Go ahead.
So I was just I guess we'll start with the gas bubble question. So I'll be really honest. Gas bubble disease, I find, is very difficult when it gets severe, to find any long-term success
Yes.
and relief. So, and I've seen a lot of attempts in various different ways of trying to deal with it, and it's just something that is recurring. Why is it recurring? It's recurring because it's facilitated by stress, and so the animal. So
Yes.
you attempt to help the animal, whether that would be by trying to, if the gas is in the tail and you can see the bubbles, you could try to gently poke them with a needle and get that gas out. That immediately, all of those actions cause stress to the animal. That's feedback, negative feedback, and it just causes more bubbles and more bubbles, and you just constantly rinse and repeat this endless process, right?
Okay, wait, hold on.
Yeah.
How often would you say, because you've had a lot of seahorses, how common do you think you would say this gas bubble disease is?
So at scale, I mean, at least from the literature, I'll give a number. It's about 20% of disease, or at least is a symptom. Is usually about what it's cited. About 20% of cases.
And you didn't let him answer my other 5 questions that
Has
I
some
asked
type
in
of gas
one.
bubble.
And I think Ray has one too. But go ahead, Dimo, and continue to talk about just, like how, if you don't like freshwater dips, what do you, because Ray said freshwater dips, in my opinion, takes care of an immediate severe problem and gives us time to remedy water conditions that cause the greater infestations in the first place. But I mean, I hate freshwater dips. I admit that publicly. I hate them. I've used them in certain circumstances, but they're awful. If the seahorse has a ciliate infestation, they are flipping around. It's awful. So, I mean, like, what would you do instead when you see these problems happening in the tank? Would you not remove the seahorses with problems into a hospital tank, or B, at least use a freshwater dip to clear them? And then what do you do to the tank? Sorry.
I agree with Ray. So it's all about the circumstance, and that's usually because I get a lot of these questions. I actually just was talking to another fellow that was seeing some stuff with his seahorses, and it's very difficult for me to provide perfect advice because I only get to see a very small snapshot of what you're
Yes.
telling me, right?
Yes.
And usually, at least novice hobbyists see a problem way too late, in the sense that we're already
Yep.
well past the preventative, the mild, and we're well into now moderate to severe disease, whether that would be gas bubble, bacterial, parasitic infections, et cetera, et cetera. So I find that the more you see it, the earlier you'll see signs, and the faster you can react. So I agree with Ray. I think that a freshwater bath is for a very, very severe case. I think it buys you maybe I think it buys you days. I don't think it buys you more than that. Um, so what my experience, and I'll speak from my experience, is I use formalin typically when it comes to external parasites like ciliates. Um, the reasoning for that is I can use it in very, very dilute concentrations directly into the environment, especially if we're talking about a sterile system. So if we're raising seahorses in a very sterile environment, I would use formalin, because it is extremely generalistic medication, and it will just wipe basically everything on every surface that it touches.
Totally agree. We actually have had this discussion before. We have talked a lot about using formalin as a preventative, even in a low dosage in fry tanks. But you're talking about, again, using formalin to treat adult seahorses in a sterile environment. So I just want to make the comment again for any Zen Gingers out there that you wouldn't want to use formalin. And can you explain a bit more about why you wouldn't want to use formalin in a tank that had sand, rock, coral, all that stuff in it?
Yeah, so formalin. I think you guys even mentioned it before. It is very, very toxic. Formalin is short for formaldehyde. Really, it's so typically what you'll find available and is about a 10% formalin, which is actually only about 4% formaldehyde. Really, so even though it's very, very diluted. It is very, very nasty stuff. It is carcinogenic, as you had mentioned. It reeks very, very strongly. It has a very, very strong smell. It should be used in well ventilated spaces. And I think it goes without saying that formalin in any, like I said, in a reef tank where you have live rock and you have sand, you have corals, et cetera, it will wipe everything. It's everything that it touches anyways. I think some part of the sand bed and maybe even some of the anaerobic bacteria within live rock might remain, but I think at least on the surface you'll cause such an incredible ammonia spike that you'll wipe everything, right? yeah, so it's not a good solution if you do not run a sterile environment. And yes, we also used at the lab formalin as a preventative for various external parasites. Yeah. And it works really, really well. It works really well.
Well, that was one question I did want to ask before we have to go, guys, because Kelly has to be up at 4:30 in the morning, and I'm sure we've taken up so much of Dimo's time. He's like, "Oh my God, I'm never coming back."
No, actually,
But
actually, I've been having a great
was
time.
like,
I really appreciate you having it.
okay, good, because we love talking to you, and this is what we do every week. We get into a conversation and we keep going and going and going, but we don't want to take up all your night. But I did want to know a little bit more, and we do still have to ask about coral. No, we asked about corals. Yes, we did. Okay, good. I wanted to ask about some of your research with the pathogens, because that's like the big thing with seahorses is we try to avoid organics, we try to avoid too much buildup. We try to make sure, even if we don't want sterile systems as hobbyists, that we're keeping them clean because we don't want the seahorses to get sick. And the thing is, seahorses are delicate creatures, but they're really strong too. Like, I mean, they're tough little broads. I mean, they can withstand things that I can't even, like, I don't see my discus freshwater fish ever handling, right? I mean, I'm just saying they're resilient, but they can get sick very quickly because of their immune systems. And I'm just curious if you could talk a little bit more about what you learned when you were researching your pathogens and how to avoid them, how to treat them, how to anything them.
What wonderful. So I'll also answer, I think, the Zen Ginger's question all at the same time. So, and I'll even go back because I still haven't said it. So what I would recommend is that you stay on top of your maintenance. I think that preventative care is the solution to almost, I will say almost, because it unfortunately not always is the case, but most instances of disease can be remedied through very, very good observations and a strong maintenance routine where you maintain your aquarium. And again, the question is, so what would you use to treat the tank before putting your seahorses back? I think that in an ideal scenario, you would siphon as much of the detritus and other buildup in the aquarium as possible. I would scrape all sides of the glass, let that all settle to the bottom, collect all of that nicely with the good siphon. That's also why I'm not a huge fan of live rock into your aquarium. It's just, unfortunately, there's no real good way of dealing with live rock. But what I recommend is, and I think I mentioned that to you, Sophie, is blow all of your rock with a turkey baster. Do not let the detritus settle on it. Use copepods. Another good reason to have get yourself a culture of like Tisbe copepods. They actually do breed in your aquariums. They will colonize your rock work. They'll keep it clean. They'll eat the detritus. So that's one way to do it. Yeah. And anyways, the pathogens from the literature and from my experience, it's most of it is bacteria related. Parasites are a very, very small fraction. They're actually roughly equivalent to mortality cases like gas bubble disease is. So they're sub 20% of mortality episodes and cases. Everything else is bacteria, and or it's a combination. This was the other thing that I was talking about with gas bubble disease. It sometimes it's just a symptom of an underlying bacterial condition. So typically, and bacteria there's hundreds of species of bacteria that are pathogenic to seahorses. Heck, some of them I don't even remember anymore, but there's so many of them. Anyways, it's so easy to write a seahorse publication, when you have a new bacteria, all you have to do is well, I'm oversimplifying, but you would identify it, grow it, you reinfect, and Bob's your uncle. You have a new paper, right? Um, but bacteria is really causes two issues. One is dermatitis, which is your external. You have an inflammation of the skin, right? So anything that ends in itis is inflammation, is what we call it, and enteritis. So you have inflammation of the intestinal system. So those are usually the two causes of mortality in seahorses, and it is almost always caused either alone by bacteria or in conjunction, bacteria in conjunction with another like a parasite, for example, right? So gas bubble disease, you'll find very commonly wherever your gas bubble is, you have a site of crazy hemorrhaging and inflammation from bacteria. And it's usually the bacteria that infects a particular region, whether that's inside the pouch, usually you see in males, or on the tails. And it's just dermatitis that's gone out of control. And then the seahorse gets super stressed out. The immune system is very poor, the response is very, very poor, and you get gas bubbles, right? Directly under the skin.
Or, and I'm interrupting, I know it, and I'm going to let you finish, but pouch emphysema too, which isn't technically gas bubble disease, but it's kind of like related. But I had a seahorse that scraped his pouch on a rock, and then all of a sudden he was floating at the top of the tank, couldn't swim down, pouch was all inflamed, and big white spot. No, Dermot, I love what you just said. All right, I just wanted to agree. Go ahead.
Yeah, so
I'm curious about with internal gas bubble disease. It's my belief that it's usually an organ problem, organ failure, or maybe it's bacteria affecting the internal organ. But the use of Diamox or acetazolamide is the standard treatment for it. Unfortunately, when I come up with the thoughts that Diamox being a diuretic, why wouldn't other diuretics work as well? But unfortunately, I never ever had an internal bubble disease after that to try it, even though I've got scans of different diuretics here that I've used for myself and my animals.
yeah,
Would have been nice. Would have been nice to try, or at least to have someone with the knowledge that could help out, because a lot of people have trouble getting Diamox, and yet with diuretics being such a frequent treatment for blood pressure disease, most people are going to know someone that has a blood pressure problem and is using a diuretic, and you could scoff a few pills from that person to get you out of the fix if it would work as well as the Diamox.
And I'm sorry that we never let you finish a question or a sentence, but now I will let you. Please continue.
No, I was just going to say I think Ray has a great point, and it's something that I really want to talk about, and that is the so I will say as of the last maybe 10 months that I have not been staying on top of the literature. I've read almost every single seahorse related scientific paper that's been published ever at this point, and I will say that none of that is published. None of what you said, Ray, is published.
Yeah, I couldn't find any of that. That's why I wanted to try it for myself. But when you don't get any seahorses with the problem, it's pretty hard to experiment for a solution.
And I think that what I also want to say is that I think that this is where hobbyists in some ways are much better at providing such guidance than the scientific community is, because we tend to as in the scientific community, unless it's a large scale problem, it's not a problem. That's usually the way that we treat things. And also, but it is also, I think, important to take something from other hobbyists with a little bit of grain of salt, and be careful about anecdotal information. I think that's
Sure.
where the hobbyist does lack, unfortunately, is that there isn't enough uh, tried and true methodology set to apply to every situation and every person that is experiencing a particular disease problem. So I think that,
But you know what's sad though is that,
yeah,
I'm sorry, I'm cutting you off again. But what's sad is that, most average hobbyists don't. Okay, so maybe they have access to, but they won't go look at scientific literature, right? And so they are getting, like Ray was talking about before we went live, the Facebook posts and such, and that's why I'm trying to create a website. That's why you're trying to create a website, etc. But the thing is, like with Diamox, I think someone like you needs to like experiment with it if you see this problem, because even though it's not hitting aquaculture large scale, it should be documented somewhere. And finding in a different route than Diamox, because most people can't just get it.
yeah, yeah. I agree, and I think that yeah, it's very very valid.
And I
No, I
cut
was
you
just gonna
off.
say
So do you even remember where you were?
it's just more
Sorry.
it's unfortunate that there's always a divide between the scientific community and the hobby,
Yes.
even though I think that both work really really well together and that they can really help each other. But yeah, it's and I think it's something that we're starting to see more of, especially if you go and look into and you're into corals and things like that. I think the fish breeding community is a little more, how do you say, closed door, I think, and it's because there is a lot of, I guess, monetary association with a lot of the details, both overcoming a particular breeding step or diseases that create bottlenecks, things like that. But you'll see that in the reefing community and in corals, that more people from the scientific community are coming and working with hobbyists and in industry, and vice
Yes.
versa. And I think that that's important, and that's really what I want to do. I think that's the to be able to in a somewhat layman fashion be able to talk to everybody, and to unite and extract information from both places and make sense.
Oh my God. Yes. 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 Love it. I love it. And we are definitely here for you. I will put you in touch with every connection, and you just got to do some
Yeah,
experiments to it for us, and you got to send us stuff to the US.
and
That's all.
there's one last thing that I will say, and I don't want to ramble too much, but
Yes.
on the topic of
No.
disease, I think that's there's two things, at least from the literature. The first one is that almost all antibiotics work to a certain extent. So, and I don't even remember all of the antibiotics that have been tried and tested, but for the majority, there's so many species of bacteria that affect seahorses that you're bound to find one that is going to be resistant, even to the most general,
Resilient.
and will not work for your issue. So, so
So what do you
I suggest,
suggest?
I'm very firm on trying to be on preventative medicine.
Proactive.
Yeah,
Yep.
and the one very interesting thing that is very much I think emerging in the literature, especially for seahorses, is preventative medicine, and particularly using probiotics, things like lactobacillus species,
Yep.
and integrating that into your
Yep.
feeding regimes, to reduce and compete with bacteria, with pathogenic bacteria,
Okay, so
and
that
raise
could
correctly.
be an entire, well, I was going to say that could be an entire another episode. You're going to have to come back so that we can talk about probiotics, because that was Dan's new thing when he left. when he had family stuff. But the thing is, I totally agree, and we all do agree. There just has never been anything. It's like everybody says, be preventative, and we try to, tell people how to be preventative, meaning set up biggest water volume you can, make sure you have a huge skimmer. I personally like UVs, some people don't, but whatever. We try to help people set up for seahorses using the best method so you don't ever have a problem, you never run into this, right? But when people do, I agree with you. Like I do have a video out there where I treated for a skin eating bacteria on a seahorse, and I used Furon 2 and I used triple sulfa, and it worked. Thank goodness for me. But then the next time I used it on different seahorses, it did not work, because I think we're creating resilient bacteria, or there's so many so many species, like you said, that our antibiotics aren't working anymore, and that should not give anybody who has a problem less hope. We will still help you try to figure it out, and we'll do what we can. But I think preventative is the answer. And I think the things that we're now talking about, like peroxide, like when you're raising fry, enriching, like making sure that the tank is adequate for the seahorses is really important. Water changes, all that stuff. I'm just so glad you're bringing up these topics from a scientific perspective, because it means we're all on the same page.
Yes, very, very much so.
So
And like I said, I think that the hobbyists, in a lot of instances, have much more knowledge, much more hands-on experience than a lot of the scientific community does. And I think that, yeah,
well,
it's particularly that hands-on experience that I cannot stress enough. I spent yeah, about 9 years in the lab, and the amount of turnover of students, which is very common. You have new semesters. Usually, a student would remain for a semester or two, maybe a year. You have some really good ones that really are very, very passionate about what you're doing, and so they stick around for a little bit longer. But the turnover rate is very high, and everybody that typically is in school has very, very little, if any, hands-on experience. It's all from scratch. Even a lot of the faculty and a lot of the professors, even my professor, who was actually my professor being taught by me and Zach about the biology, the husbandry, all of the little everything that we do as hobbyists, right? And I think that yeah, it's really, really handy when you're both a hobbyist and you want to learn the science behind it. It's that's when it becomes really, really interesting.
Well, just keep that in mind when you get, all famous, because you're some famous scientist. But in the meantime, we are going to need you to start shipping to the US. We've got a list here. We're going to need you to come back. We're going to need
Yes,
your partner
yes.
to come back, and I would say too that just, it's about networking, right? Like it's about, me putting you in touch with my contacts, you telling maybe your people, hey, maybe listen to these guys. They've been doing this for years and years and years, and they figured something out, right? Peroxide works in the hatchery. This works in this, and but I agree with everything you said tonight. I think this has been amazing. I'm over talking Sophie. I'm sorry, Sophie. You know me. You should just kick me out when you're running a session. But if there are any more questions, ask them now. We will have Dimo back. Zen Ginger is begging, and we will have you back. We want you and your partner to come in. That'd be amazing if you guys could pull that off. But in the meantime, let's connect on the side.
Absolutely.
But I'm giving it to you, Sophie. Were there more questions that anybody had? I have like 500 more about pathogens, but I'm not going to ask
I
them right
say
now.
Dimo
Go ahead,
has to
Sophie.
come back and we can do maybe a more narrow topic each time, like one about the
Yes.
pathogens, one about this.
Yes. Yes, because this was just
It was
introducing him to the
wonderful. Thank
channel,
you so
and now
much.
we got to like go into we gotta promise him it's only going to be an hour instead of 2.18, and to not take up all his time, but also a specific topic about, we're going to talk about the pathogens. What do you do about
Yeah,
this?
what did you
What do you think about
find
that?
and that
That'd
kind
be
of thing.
amazing.
Yeah. Love that.
I mean, your scientific experience and knowledge is just crucial to us, because you're learning stuff we couldn't even know, ever. So we appreciate you. Sophie, take it over.
Well, I don't have much to say. We don't have any more questions, but I can vouch for the fact that he breeds good seahorses. My seahorses are doing very well.
Yay!
I love them. And yeah, I did get fry for them, so they're very good seahorses.
I think
We
that's
definitely need to have you in again. I think Kelly will have you in every week if she gets her way.
Yeah.
Yes.
But one second.
yeah. I was just gonna say, let the animals talk. I think that's all that. Yeah, I mean, there's a lot for us to talk about as well, but yeah, it's important to maintain focus on our animals. I think more than anything. Yeah, they can teach us a lot.
Absolutely.
We can teach them a lot as well, and we can learn a lot from them and share between each other. But I think the number one thing is, I think that we should really, because I think initially, especially with something like a seahorse, it's so surreal that you're able to maintain it at home. And then I think because we have to feed them so frequently, it becomes very routine and monotonous. And that is when we become, I think, complacent, and we start to miss things. And I always tell everybody and I do so many feeds every single day to so many aquariums, and the number one thing that I think that you should do is focus on the task at hand when you're doing it. And I think pay close attention to what you're doing. So if you're feeding your animals, make sure that you're really engaged at whenever possible. Yeah.
Hey, we thank you so much for coming. I'm sorry I can't, say thank you to your face because I'm pink and green. Here we go. Let's try again. Nope, I'm pink and green. Okay. Next week I won't be. Holly Ray, I'm sorry if I didn't let you ask him, or I'm sorry if Sophie and I didn't let you ask him all your questions.
I
But
did
we
fine.
will absolutely have him back. I'll beg, plead, borrow, and make sure he gets back in here, because this has been so much fun
Yes.
and so interesting. So yeah, we're gonna have you back, and we're gonna have your partner. But Sophie, go ahead.
All right, so we'll finish it off for now. Thank you for coming. This was really interesting. Definitely a different point of view. And we'll see everyone next week. All right, happy Wine Wednesday, everyone.
Cheers.
I'm raising my glass.